Right, so we're going to continue talking about our innate non-specific host defenses. This is going to be for 17.4 and 17.5 together. So the first thing we're going to talk about is pathogen recognition and phagocytosis. And we've talked a little bit about these things, but we're going to kind of put them all together now here. Starting with what's called extravasation, also called diapedesis. So this is when our white blood cells are going to pass through the walls of the capillaries and then migrate within the tissues to the site of infection or damage. So we have spoken a bit about this already. If we, for example, cut our arm or a hand or something, we have a cut, and let's pretend when that cut happened, it brought bacteria into the body. So we have a cut, and then the bacteria is in the body, but then also we have all of this cellular debris, we have cut capillaries, and things like that as well. So those cut capillaries, the destroyed self cells, and the bacteria are all going to cause some things to happen. So this diapedesis is going to be initiated by, for one, the complement factor C5a, which if we remember back to our complement system, this is one of those in that kind of pathway, the complement system, and cytokines. So getting back to our example, when we accidentally cut our skin, for example, the resident macrophages of that tissue, so those that are in the skin in this example, and the damaged tissue cells themselves are going to end up releasing those cytokines. Remember, those are chemical messengers. So they release cytokines, those chemical messengers, and we also have the C5a being released. That is going to initiate inflammation. Remember, C5a is part of the complement system. A lot of those initiate inflammation. So it initiates inflammation, and then our chemotactic, which is just a way of saying that they go through chemotaxis. So remember, chemotaxis is the movement in relationship to chemicals. So they are chemotactic, meaning that they do move in relationship to chemicals. In this case, what we're talking about are cytokines and C5a. So the cytokines are going to affect the blood cell walls, and then what this does is it upregulates adhesion molecules. So these cytokines that are released by the resident macrophages and those damaged tissue cells, those chemicals are going to move through the tissue. They're going to get to the blood vessels that are close by, so those capillaries nearby. It's going to affect those blood cell walls, so the actual cells of the capillaries, and make them stickier. So upregulating adhesion molecules means that it's going to make those walls of the blood vessels stickier. So the white blood vessel, or the white blood cells, are then going to stick more via these adhesion molecules when they're floating by in the blood. So if we have our vessel here, and then we make this area a little bit stickier, when we have our white blood cells coming along and it bumps into the side here, rather than maybe bumping back off, instead it's going to stay stuck to the side here. And it's actually going to slow them down. So they're going to be rolling along the side here because they're kind of sticking to these adhesion molecules, slowing them down rather than going with the flow of blood that's continuing. So it slows them down, and then when they slow down enough and they reach a cell junction, then they can move through those cells. So they can move in between the two cells to get to the site of damage. So if these are individual cells, which of course they are, if we're talking about our blood vessels, then this cell, once it gets here, it can move in between these cells here. So part of our response, part of our inflammatory response is that our blood vessels will dilate, and they also become more leaky. And since they dilate and they become more leaky, then there are larger gaps in between these vessels. So that's the leaky part. Remember, it's going to cause edema. We release more fluid, so fluid's coming through those blood vessels, through the cells in the blood vessel walls. And then also these white blood cells are going to be able to move through there. Then we have transendothelial migration, which means that they are flattening out and squeezing through those cellular junctions. So they're going to trans, meaning moving through, and then the endothelial cells, and they are migrating. So they're migrating through the endothelial cells. So they kind of flatten out and squish and squeeze and make their way in between all of the cells that then would be here until they get to the site of damage. So this is diapedesis, right? So diapedesis is this movement, these white blood cells that are passing through the walls of the capillary and then migrate to the tissues, or migrate through the tissues to the site of infection. Again, as we said, our neutrophils are the first ones to arrive, and this happens within hours of a bacterial infection. So we have that cut, the bacteria in the cut. First of all, our body is going to go into response mode here, so it's going to start to repair the area via our clotting mechanisms, which we'll talk about in just a moment, and then initiates inflammation, sends out those cytokines, and then we have the neutrophils being the first ones to arrive to the scene within hours of this infection. Then we have the monocytes, which are going to take days to get there. So they'll migrate to the tissue, and then they'll differentiate into the tissues, into macrophages that are specific to that tissue, and then they're going to continue to help the neutrophils, which, remember, what the neutrophils and the other monocytes do is that they are phagocytic cells. So they're going to be gobbling up cellular debris, they're going to be gobbling up bacteria that they find in the area because of this cut, and because an infection is forming in this cut because we have bacteria there. So this is an illustration from the text of exactly what we just spoke about. So in this case, you can see this is our blood vessel, this is our cut that we were talking about. We have pathogens that have gotten in. Here's our neutrophil, the first on the site. And then you can see these are our capillary epithelial cells. All these little dots that are in here, these are all of those cytokines that are being released in the area. This is our resident macrophage here. So it is, you can see it's engulfing this pathogen right now, and it's releasing some of these cytokines in that C5a. So it's releasing all of these things, making, again, the bloodstream here, the blood vessel walls more sticky, and you can actually see that they're now moving through, they're emigrating to the site of injury and infection. So they're squeezing between those capillary wall cells, and they're going to migrate along here to the site of infection so they can start to gobble everything up. And then also we have the neutrophils. Remember, the neutrophils release those cytotoxic chemicals. So they are going to be phagocytic and gobble up things, but they also are going to be able to help kill those bacteria as well by releasing their granules into the tissue. And you can see this macrophage is still gobbling up as well, which will continue releasing cytokines, continue releasing the pro-inflammatory molecules so that they continue to have chemotaxis occurring. So bringing more and more white blood cells to the area to clean up this mess. All right, so how do these cells know when we have bad guys? Well, one, we already talked about those MHC proteins. So we have our cells that are moving along, taking note of the MHC proteins on the surface of the cells, saying, you know, this is self, okay, this is self, okay, and then saying this is not self, this is not okay, and we need to do something about it. We also have other ways to recognize pathogens rather than just the not self. We also have some other things we're going to talk about. So the first one is pathogen-associated molecular patterns, or PAMPs. So these are molecular structures, as their name implies, that are common to many groups of pathogenic microbes. So these are specifically recognized by pathogenic cells. So again, pathogen-associated, so associated only with pathogens, and then these are specific molecules that are in a particular pattern on a pathogen that we don't find in our own cells. So for example, peptidoglycan, right? So peptidoglycan is a molecule. It's a molecule that we don't see on our own cells, so then it's associated with a pathogen because our cells don't have cell walls that contain peptidoglycan because they don't have cell walls at all. And so any molecule that's associated specifically with a cell wall is going to be a PAMP, a pathogen-associated molecular pattern. Another example is flagellin, so a protein that's in flagella, and this is because within humans there's only one cell that has a flagella, and that's the sperm cell. And the sperm cell is actually sequestered away from the rest of the body, away from the immune system, so that the immune system doesn't actually attack it because it is a PAMP. Well, not the entire thing, but the flagellin that's within the protein, or the protein flagellin that's within the flagella. Is a pathogen-associated molecular pattern. So when one of our immune system cells, one of our phagocytic cells, finds this, it's going to recognize that it's not self, it's a pathogen. Another one, lipopolysaccharide. So this is, again, remember the outer membrane of the gram-negative bacteria has this molecule, and we've already spoken a lot about it. But again, this is a PAMP, so something that our body recognizes that is not self, that is associated with the pathogen. And then also lipopeptides. These are expressed by most bacteria. So lipo meaning lipid, and then peptide meaning proteins. So lipid protein combinations that are expressed by bacteria. Again, we don't express these, so they're not self. And then DNA or RNA. So our cells are going to recognize that if it finds any DNA out there, that there shouldn't be DNA out there, because our DNA stays within our nucleus and we like it that way. So if it comes upon DNA or RNA, something's wrong, and it can go about fixing the situation. So along with PAMPs, we also have pattern recognition receptors, or PRRs. So these are structures that allow phagocytic cells to detect the PAMPs. So again, as these cells are going along, and they're testing, is this self, is this self, is this self, then we have these PRRs that are on the phagocytic cells themselves, and those are the things that are going to be recognizing these pathogen-associated molecular patterns. So an example are toll-like receptors, or TLRs. So our toll-like receptors, so again, these are recognition receptors, so receptors that act like a toll. So they bind to various PAMPs, and then they communicate with the nucleus of the phagocyte to elicit a response. So it's like a toll. It connects to the PAMP. Once it connects to the PAMP, then that sends a message. This receptor is going to send a message to the nucleus of the phagocyte, and then that's going to initiate a response. So that's going to tell the phagocytic cell to then start to engulf the pathogen, and then degrade it and make enzymes to degrade it and things like that. So most of these PRRs, these recognition receptors, are located on the surface of our cells, so on the surface of the cell. But some can be embedded in the membranes of organelles within the cell. And this is important for pathogens that have entered the cell prior to phagocytosis taking place. So for example, our viruses. So if a virus allows itself to get brought into the cell, so think of HIV, for example. HIV tricks our immune system cells to get itself into the immune system by attaching to our CD4 receptor, and then that CCR5 receptor brings that virus into the cell. Once that virus is inside the cell, it hijacks things, inputs its DNA into our DNA, and then gets to make lots and lots of copies of itself. So in the inside of our cells, we have some of these receptors, these PRRs, so that they can bind to different pathogens within the cell. So if we somehow get a pathogen inside of the cell, we still have the ability to have this matchup between the PAMP and the PRR so that we can still have some sort of cellular response, even though the pathogen has already gotten into the cell at this point. So most of them, however, are on the surface of the cell in the plasma membrane, because hopefully they haven't been infected by something already, by a pathogen, and so they're outside of the cell, and they're going to be interacting with the PAMPs that way. So you can see an image on the right here of kind of what we've been talking about, this interaction between the PAMPs and the PRRs on macrophages is going to provide an activation of the macrophage. So as we said, when these cells are activated, then they're going to produce and secrete cytokines and enhance intracellular killing. So if we look over here on the right, we have a lipopeptide. Like we said, that's a PAMP. We have flagellin. We said that was a PAMP, something that we find in flagella. And then peptidoglycan here. Remember, that's a PAMP as well because it's on a cell wall and we don't have those. So we have specific PRRs that are peptidoglycan receptors, PRR that are flagellin receptors, or PRRs that are lipopeptide receptors. So that when that lipopeptide clicks into place, or the flagellin, or the peptidoglycan, then it's going to send messages within the cell to the nucleus. So then when this happens, it's going to have the effect where it's going to produce and then secrete these antiviral interferons and other cytokines. So it's going to start to release those chemical messengers in order to pass the information along to others. Just like we saw with the illustration with diapedesis, if we have a macrophage in the area where the cut has happened, this is occurring. So we have the pathogen is inside the body at this point, it's recognized, and then we have the release of these cytokines. You can see here that we also have in the inside, this is an example of a phagosome here where the pathogen is inside of the cell and it has a particular nucleic acid, for example, and then there's the nucleic acid receptor. So again, this is a PAMP, this right here is a PRR, and then it's going to still communicate with the nucleus and then produce these antiviral interferons and cytokines. So PRRs respond to chemical distress signals from damaged or stressed cells. And then this is going to allow for assistance in inflammatory response from injury rather than only in response to infection. So what we're saying is that even if we have cells that are in distress, they're damaged, they're stressed out, they're getting too old, things like that, our PRRs are going to respond to that. And then it's going to assist in the inflammatory response in response to these stressed out cells or damaged cells from injury rather than only in response to the infection. Meaning that if we cut ourselves and say we don't get a pathogen in the body, say there was no bacteria on the knife or something that we accidentally cut ourselves with, so then these cells are still going to respond to distress signals. So the cells that were cut are sending out those chemicals that are communicating like, hey, there's a problem here, and then these cells can initiate or can respond accordingly. So phagocytosis, if you're not familiar with that, we're going to go through that in a little bit more detail here. So we've already talked about how it is engulfment of the pathogen, and then it is brought into a vesicle, into the cell. What it does is it forms these pseudopods that are going to wrap around the pathogen. And we kind of saw that we had our cell earlier, and then we had our pathogen, and then we could kind of see that we create these pseudopods that go around the pathogen. And then these are going to continue, kind of like hugging it, continue around until they form an actual vesicle and bring this pathogen into the cell. This will be the nucleus here. So then it pinches it off, as I kind of just drew here, where we have those pseudopods that kind of come around, and then they eventually connect. And remember, all of this is made out of plasma membrane, so once they connect to each other with the plasma membrane, then the plasma membrane that was in the inside here encloses this bacterial pathogen. When this happens, this now is called a phagosome. What the bacteria is in, what the pathogen is in, is a phagosome, and then it decreases the pH. So it becomes very acidic, pH 4 to 5. So that decreasing of the pH, one, it'll kill pathogens that can't be in a more acidic environment, but it also makes them weaker. So even if it doesn't kill them right away, it will make them weaker so that we can utilize other things, meaning that this phagosome itself will then fuse with one or more lysosomes. So if this is our lysosome here, all of these dots are our enzymes, which is lysozyme. So this is our lysosome. I can just do this. This is our lysosome. This is our phagosome. But then when they merge, so we're going to merge both of these here, and now they've become one giant vesicle, and that giant vesicle is now called a phagolysosome because we have the phagosome and the lysosome together. Now when this happens, it's going to enhance or increase acidification. So again, keep bringing that pH down so that it eventually kills the pathogen or weakens the pathogen enough to where we can digest it in other ways. So it activates our pH-dependent digestive lysosomal enzymes. So within this lysosome here, remember the dots were our enzymes, our lysozyme. Now lysozyme is going to be activated by a decrease in pH. So once that decrease in pH happens, it starts to activate those enzymes, and those enzymes can go in and start to break apart the pathogen. Also, we have production of hydrogen peroxide and reactive oxygen species. So we make hydrogen peroxide, we make reactive oxygen species, and all of these are going to start to attack the pathogen. So these are things that are very dangerous to a lot of pathogens, and so they work to just kind of start to break apart their cell wall and their plasma membrane and break them down so that we can digest them. So these activation, or these enzymes that were activated up here, these pH-dependent digestive lysosomal enzymes. So I mentioned that one of them is lysozyme. So when we're thinking about lysosomes and therefore phagolysosomes, then we're talking about a decrease in the pH, Once that decrease in the pH happens, then that's going to activate things like lysozyme, also phospholipase and proteases. So we're going to be breaking apart phospholipids. We'll be breaking apart proteins. Remember, that's a huge part of our plasma membrane. And then we have lysozyme that's going to be breaking things down as well. So then when we are talking about these phagocytic cells, what happens when they are trying to go through the process of taking out some bacteria, they have what is called a respiratory burst. So this respiratory burst is when these phagocytic cells are going to increase their uptake of oxygen. So this uptake of oxygen is important because it does things like increases the production of the superoxide anion. So remember, that's a reactive oxygen species. We just said that it's going to utilize those reactive oxygen species, activate those to try to destroy the pathogen. So in order to make these reactive oxygen species, like the superoxide anion, as well as hydrogen peroxide and our hydroxyl radicals, other reactive oxygen species, we have that decrease in pH, and that's going to, as we mentioned just a moment ago, we have the decrease in pH which activates these things, but in order to have these things, we need to have the oxygen to make these things, which makes them reactive oxygen species. So we have this respiratory burst. So the phagocytic cell is going to have this burst, this increase in uptake of oxygen, so we need lots of oxygen, and then it's going to be able to produce these things that are going to start to destroy the bacteria. So we also have reactive nitrogen compounds with cytotoxic potential are going to form as well. So we have this respiratory burst, meaning increase in oxygen, which for the most part we're talking about our production of all of these things that contain oxygen, because remember they're antibacterial. But we also have reactive nitrogen compounds. And then these are also cytotoxic, meaning toxic to cells, and then they can help destroy the pathogen. It also can release a storehouse of defensins. So remember, defensin proteins are going to destroy pathogens outside of the cell also. So we can release these defensins because if the phagocytic cell is busy right now creating this phagolysosome and then trying to destroy it and break down the pathogen, it can also send out and release these defensin proteins so that it can start to destroy pathogens outside of itself while it's working on gobbling up the pathogen it just grabbed. So then the remains of the pathogen, so remember we grabbed onto the pathogen, we brought it into a phagosome, merged it with a lysosome, and now we have phagolysosome. We're breaking these things down with these reactive oxygen species. Then the remains of that pathogen are going to be processed within that phagocytic cell, and then we're going to take key antigens and present them on the surface of the macrophage. So we're going to take parts of this bacteria. So the macrophage has gone in, destroyed this bacterial pathogen, broken it down into its pieces. Then it's going to take its pieces, specific antigens, and it's actually going to present them on the surface. Basically saying, hey guys, look, I got this guy. I broke him down. This is what he looks like. And now all of you other cells that are coming around need to know what the bad guy looks like. And this is what it is. Now, we're going to talk about this in a lot more detail in Chapter 18, so this is part of the adaptive immune response, and we'll talk about it then. Then what we have is leftover waste products, so those that we don't present on the surface of the macrophage, we have kind of all this leftover stuff, all this other leftover stuff, and these are going to be excreted from the cell in an exocytic vesicle. So we secrete them from the cell, and they can go to various areas and be recycled or be destroyed or just be released as waste. So, via our typical waste things like urine, feces, sweat, and we can get rid of these extra things. So here we have what we just discussed in illustration form with our bacterium here coming in a phagosome, which is the next step here, the phagosome. Then we have our lysosome that contains our digestive enzymes going to merge, and it becomes a phagolysosome. And then we have our undigested waste, and that's going to be released to the exterior via this via exocytosis. All right, so we have spoken a lot about inflammation. So now we're going to go into a little bit more detail about inflammation, what it does, why it's important, why we want it to a degree, and what it does for the body. So inflammation is triggered by a cascade of chemical mediators as well as cellular responses, which we know, right, because we've already talked about a lot of these chemical mediators, all of these different intermediate proteins in our complement system, so those different types of fragments that are going to initiate or trigger inflammation, and then other cellular responses. So inflammation occurs when cells are damaged and stressed, or when pathogens have breached the physical barriers. Right, so if our cells are damaged in some way, so say we bruise, we bump into something and we bruise ourselves, then we have some inflammation. If our cells are stressed, if we've kind of used them too much, or think of something like maybe blisters, for example, we've stressed the cells, we've kind of destroyed the area. And then when pathogens, of course, have come in, they've breached our physical barriers, they've gotten past the skin or mucous membranes, and so then now we have this inflammation triggered. So it allows for the recruitment of cellular defenses. So inflammation is important because it is going to go get other cells to help defend the body and get rid of the pathogen. It helps for the removal of damaged and dead cells. And then it also initiates repair. So it's going to help to get rid of those cells and then start repairing what's left. However, excessive inflammation can result in local tissue damage, and it can become deadly. So that's why I continue to say, like, inflammation is good. We want inflammation. It's an important process. However, if it gets to be excessive, if it gets extreme, then it can cause some local tissue damage and become deadly. So you would want to get to a doctor or a hospital if that's the case. However, what we are often talking about is either acute inflammation or chronic inflammation. So acute inflammation is early or immediate response to injury. So again, if we cut ourselves, then right away we're going to have all of those things we talked about. The cytokines are released, diapedesis, all of these things. Our capillaries become leaky, etc. That is acute inflammation. So it's immediate or early response to injury. And then we also have chronic inflammation. So this is an ongoing low-level battle between pathogen and host. And then this is when our acute inflammation is unable to clear pathogens. So we see chronic inflammation when we have some of these diseases that can last a really long time. So, for example, with HIV or Lyme's disease or hepatitis, things that last for a really long time, we can end up seeing this chronic inflammation. And that's because there's this low-level battle happening. And that's because we have a pathogen in the body. Our body is kind of low-level trying to get rid of it. And then this can continue on for years or lifetimes. And this, of course, is when our initial or acute inflammation isn't able to clear the pathogen. So when a person does get HIV, at first there's an acute inflammation or an acute inflammatory response to try to clear the pathogen. But then for the rest of the person's life, then they have this kind of low-level chronic inflammation. So let's take a look at how inflammation happens, what the events of inflammation are. So first we know that there's an injury. So an injury occurs, we cut ourselves. The very first thing that happens after the injury is that we have vasoconstriction of the blood vessels, so to minimize blood flow. So of course, if we get a big gaping wound, we accidentally, bump into something incredibly sharp on our leg, let's say, and it slices the majority of our lower leg. That is a giant wound, and so immediately our body's going to go into vasoconstriction mode, where all of those vessels in the leg are going to constrict, which means to become smaller in diameter. Vaso meaning vessels, and then constriction, so becoming smaller, to help prevent the blood loss so that we don't lose too much blood and pass out or perhaps die. So this is related to the amount of vascular injury, but it's usually pretty brief. So it's usually just like a quick response like, hey, let's try to decrease the blood flow here to try to prevent us from bleeding out altogether. And again, this is related to the amount of vascular injury. So if we're talking about like we cut our finger while cutting something, We are going to have some vasoconstriction of those blood vessels that are closest to the injury, but it's not going to be incredibly extreme. It's not going to be life-threatening, for example. At this time, we have histamine being released from the tissue cells. So those cells that have been destroyed are going to be releasing histamine. And then also from the mast cells, remember those are kind of hanging out in areas that are near the exterior. And then basophils. So any basophils that we have near the injury, remember basophils are actually in the bloodstream. So when we have injury to a blood vessel, so a capillary, then we're going to have basophils perhaps come by and be near the injury, and they'll start to release histamine. And then the mast cells are in the tissues. And these mast cells in the tissues are going to be releasing histamine as well, and then the injured cells. Also, we can release leukotrienes, kinins like bradykinin, and then prostaglandins as well. So all of these different chemicals that are going to initiate inflammation. So then, after our vasoconstriction, right, so immediately we're going to say like, whoa, we want to make sure that we keep the blood in our body and we don't die. So we're going to decrease the size of our blood vessels in the area. Right after that, then we have vasodilation. So this may be a little bit counterintuitive because right away we say we don't want to lose blood, so we make our blood vessels smaller. But then after that, we say, oh, let's open them up again. But that's important because we want to dilate them or open them up so that they have a larger diameter. And this is because we want more blood flow to the area so that we can bring the things that will help to protect the area. So not only do we have vasodilation, where we want an increased blood flow to the injury area, we also have increased vascular permeability. So permeability meaning the ability for things to permeate or get through the vessels, vascular permeability. So as we are increasing the size of our blood vessels in the area, that's going to increase the amount of blood that gets there. And then when the blood is getting there, we have this vascular permeability, which can be seen as kind of like a leaky blood vessel. So we have these leaky blood vessels that are going to be then leaking this blood plasma into the tissues. And then also along with that is going to be moving those white blood cells into the tissues as well. Again, this is due to inflammatory chemicals that we've been speaking about over the course of this chapter. And then this is going to help to dilute toxins and bacterial products at the site. So again, if we have these leaky blood vessels, then we have edema. We have all of this blood plasma leaking into the tissues. Not only is it bringing white blood cells, but it's also washing things away. So it's going to dilute toxins, kind of wash those away, and dilute anything else that the bacteria is going to be producing. So think of some of those exoenzymes. So a bacteria might have phospholipases on its exterior that it's producing. And if we have a lot of blood plasma to the area, maybe we're washing away some of those enzymes. If we're washing away some of those enzymes, then those cells have a lesser chance of getting into other cells or into cells to begin with. So then, as we said, we have these leaky blood vessels. That means that our leukocytes, our white blood cells, are able to squeeze through those endothelial cells in the capillaries. And that's this image again at the bottom, is that we have our pathogens that just got in through our cut. Our mast cells that are hanging out in the area start to release histamine. That histamine is going to cause vasodilation, an increase, so an opening up of the blood vessels. And then it's going to make them leaky. So in between the cells, we're able to not only get the histamine into the bloodstream, but we have a lot of fluid that can be leaving the bloodstream as well. And you can see that over here on the right. This is kind of all of this blood that's coming to the area. And this is important because, as you can see right here, we have the diapedesis here of our phagocytic cells. Our phagocytic cells are now at the site of infection. So remember, first that would be neutrophils, then our monocytes. So all of those are at the site of the infection at this point, leaky, and then we have all of this fluid as well. That's going to be washing away different chemicals that are produced by the bacteria. So our leukocytes migrate to the site of injury. Again, remember that's chemotaxis. So chemotaxis is when cells are going to move based on chemicals. The whole process here that we're talking about is the movement of cells and them squeezing through the capillary, or in between the two cells in the capillaries, and things is called diapedesis, is cells moving. So neutrophils are going to phagocytize bacteria and then also degranulate. So remember, they can release those cytotoxic granules. Then macrophages are going to also phagocytize bacteria. They're also going to gobble up some dead cells. So remember, when you have this injury here, we've got, chunks of our own dead cells in the area. So it's going to start to phagocytize that as well. And then debris. And this is going to be part of pus. Remember, pus has a lot of cellular debris, some dead white cells, white blood cells, meaning some dead local cells and pathogens and things that have been destroyed as well. The macrophages are also going to release those pro-inflammatory chemicals. So it's going to continue with the inflammation, which means it's going to continue with making those capillaries more dilated, making those capillaries leaky, bringing more fluid to the area, which also is bringing more of our immune system to the area to help fight these bad guys and repair our damaged skin. They are activated by PAMPs or opsonins, Remember, opsonins were produced and they are able to coat a pathogen, so it makes it easier for our phagocytic cells to grab onto them and then gobble them up. Remember, PAMPs are pathogen-associated molecular patterns, so things that are associated with pathogens. And these two things are going to activate those macrophages to do what they do. So they're going to start to gobble up a pathogen because they recognize it as a pathogen due to these PAMPs, for example, or because it's coated in opsonins. So there are 5 observable signs of inflammatory response, or that one is going through or has inflammation. The first is redness or erythema. And this is because we have an increased blood flow, right? So we've dilated all the blood vessels in the area. And when we dilate all the blood vessels in the area, not only is this going to bring our white blood cells, but it brings all of the other cells in the blood, right? Which includes our red blood cells, our erythrocytes. So our red blood cells are red, and so the area has redness to it because we have an increase in the blood flow. We also have swelling. So this is our first sign, is redness. 2 is swelling or edema. We've spoken a lot about that. That's because of all the fluid coming out of those leaky capillaries. We have heat. The reason we have heat is, again, because of our blood going to the area. Our blood is quite warm. In anatomy and physiology, you perhaps have talked about, or will if you take it, or if you don't, that's fine, have talked about how our body responds to temperature, and it responds to temperature in relationship to blood flow. So we can dilate our vessels or constrict our blood vessels depending on the particular temperature, and that's related to the fact that our blood is quite warm. And so it can either increase the heat, or if we constrict it, it can decrease it in areas of our body. So in this case, of course, we have dilated our blood vessels. So our third observable sign for inflammatory response is heat. Number 4 is pain. And that, of course, is because of all of these other things here. If we have increased or dilated our blood vessels, we have all this increased fluid to the area. All this increased fluid is pushing on all of our pain sensors. It's pushing on our pain sensors because that is something that's painful. That's how they react, is by being pushed. Or when we have the cut, that cut could have pushed up against one of our pain sensors as well. And then we have altered function. So altered function means that this area is so red and swollen, and there's pain and heat, and this is meaning that we are not able to use that particular portion of our body as easily. So it's altered function. So if it's on a knuckle, for example, let's just think about that. If we get a cut on our knuckle and all of this is happening, we have an inflammatory response happening, then we wouldn't be able to bend our finger easily because we have this swelling, because we have this heat and this pain. So we have this altered function as well. So when we talk about chronic inflammation, this, again, is our ongoing low-level battle between the pathogen and the host. And then this is when our acute inflammation can't clear the pathogen. So in this case, our wound may have healed. So if we're not talking about HIV, for example, or hepatitis or Lyme disease, let's say our wound, we got some particular bacteria in our wound. We already went through our acute inflammation, but then it has healed. and the pathogen is still present in deeper tissue. So that pathogen got even deeper into the body, and then we have chronic inflammation because we weren't quite able to clear it, so we continue on having more chronic or longer inflammation. This has been associated with or involved in the progression of degenerative neurological diseases. So things like Alzheimer's, they think, is related to inflammation. So increased inflammation, meaning increased stress. So remember, if our cells are stressed, meaning our bodies, our brains are causing us to be stressed, this can cause inflammation, a low level or chronic inflammation in the body. And they can see a relationship between low level or chronic inflammation in the body and these neurological diseases like Alzheimer's and Parkinson's. It also can be related to heart disease and metastatic cancer. Metastatic means that it has moved from one location to another location. So this could be in response to a body that is inflamed or as inflammation. When we do have this chronic inflammation, sometimes this can lead to the formation of what are called granulomas. So you can see in the image down here, these are granulomas, and these are pockets of infected tissue, and they get walled off and surrounded by white blood cells. So when the white blood cells are trying to fight off a bacteria, then we can actually have these pockets of tissue that have lots and lots and lots of infection happening. So they have lots of bacteria that are reproducing in that area. And then they end up being walled off by these white blood cells. So the white blood cells come along like they're expected to when there's a pathogen and lots of pathogens. And then these white blood cells come and they're trying to get rid of them. However, the phagocytes wage an unsuccessful battle to eliminate the granulomas. So if we have this pocket of tissue where we have lots of infection happening, it can be kind of separated and surrounded by white blood cells, and those white blood cells just can't handle the infection. So it's an unsuccessful battle. They can't get rid of the granuloma. An example of this is TB. So this is why this one's so dangerous, why tuberculosis is so dangerous. One of the reasons is because we end up forming granulomas in lung tissue. So we end up having these lumps of bacteria that are infecting and making many copies of themselves, and we're just not able to clear it. And then, of course, that would lead to difficulties breathing, etc. So this can be an important cause of tissue damage from viral infections. So, for example, scarring from hepatitis C infection or liver cirrhosis. So if we think about our liver is never quite the same when we have a hepatitis C infection or if we have cirrhosis, it's in large part due to these granulomas. So this infected clump of cells that don't work anymore as regular liver cells. We have this problem that's happening. So another thing that goes along with the inflammatory response is a fever. So when we have an inflammatory response that extends beyond the site of infection, so it's not just this one little cut that we got. If we end up having an inflammatory response that goes beyond that, so the bacteria got into the body and it started to spread throughout the body, and now we have inflammation all over the place, not just this little kind of lump that's annoying on our finger where we have an infection. This results in an overall increase in body temperature. So when we have an infection throughout the body, we have inflammation throughout the body, then we end up having an increase in body temperature. The way this works is that our hypothalamus, the section of the brain that regulates body temperature, is going to be adjusted. So we're going to adjust that thermostat, which is in the hypothalamus. So certain bacterial or viral infections can result in the production of what are called pyrogens. So pyrogens are chemicals that can alter the thermostat settings. So remember that the hypothalamus is our thermostat. That thermostat, typically what it's doing is it's regulating the body, keeping it at our nice, 98.7 degrees Fahrenheit-ish, whatever's kind of normal for the body. But then when we have a particular bacterial infection or a viral infection, our cells will start to release pyrogens. And these pyrogens are basically being sent out, going to the hypothalamus and saying, we need to adjust the temperature here because we've got a bacterial infection or a viral infection and we need to get rid of it. So it's going to alter those thermostat settings and ultimately increase the temperature of the body. So it's going to elevate the body temperature, and that causes fever. The pyrogens can be exogenous or endogenous. So again, that means that within our own cells we can make the pyrogens, or part of our microbiome can start making those pyrogens, saying, hey, we're in trouble here. It's going to make those pyrogens and say we need to increase the temperature. So for example, our lipopolysaccharides, or LPS, of our gram-negative bacteria is a pyrogen. So this is going to induce leukocytes to release endogenous pyrogens. So if we have an infection with a gram-negative bacteria, the gram-negative bacteria itself has the pyrogen, and then that can induce our white blood cells to then release their endogenous pyrogens. So things like interleukins, interferons, and other ones. Then this cascade is going to lead to the release of prostaglandin from other cells, and then this is going to lead to the resetting of the hypothalamus. So we have our gram-negative bacteria is going to use its lipopolysaccharide as a pyrogen. That tells our white blood cells to release their pyrogens, so interleukins, interferons, etc. And then this kind of cascade is going to lead to the release of prostaglandins, and then the prostaglandins are going to lead to the resetting of the hypothalamus. So fever, just like some of the other things we've spoken about in this class, like pus, for example, and not washing your body, is important to the health of our body. So our functions of fever, one, it's going to enhance our innate immune defenses, and it does this because it stimulates our leukocytes to kill pathogens. So an increase in the temperature of the body actually makes it so that our cells function better. Specifically our white blood cells. So you can kind of think of this like in the summertime, we're a little bit more active, for example, than we are in the wintertime in general. So when it's nice and it's warm outside, we have an increase in temperature. Us as humans, we tend to be more active in that time frame versus when it's colder in the winter, we tend to be less active. And we can think of that in relationship to our innate immune defenses. So our leukocytes, our white blood cells, if they end up being warmer, they're going to do a better job. They're a little bit more active than if it's colder, which would be, for example, our regular 98.7 degrees Fahrenheit. It can also inhibit the growth of many pathogens. This, however, is something that is much lesser. So the main function is it's going to make our immune system work better. It's not that we can take a lot of the pathogens out of their temperature zones. However, we can inhibit the growth or slow the growth of pathogens, specifically those that are mesophiles, right? Their optimum growth is around 35 degrees C or 95 degrees Fahrenheit. So if we bring our temperature up to, say, 103 degrees Fahrenheit, we're going to be taking them out of their comfort range. So they're not going to be functioning as well. Now, at this point, we've also increased our white blood cell functions. So not only have we weakened these pathogens a little bit, not necessarily destroying them. So I want to get the point across that fever does not increase our temperature so much that we're just destroying these cells. But it can inhibit the growth of the cells. They're not as happy when we get into these higher temperatures. They're not going to reproduce as quickly as they would, or if at all, if they were in their typical 95 degrees or closer to our 98.6 degrees. It also stimulates the release of our iron sequestering compounds by the liver. So if we think of all of our ferrins or ferritins, those iron sequestering compounds are going to be released when we increase our temperature of our body. And then remember that that's going to bind up iron, and then it starves the pathogen because they need iron in order to function, in order to reproduce. So in 3 different ways, fever is very helpful. So again, enhances our immune system defense, so our cells work better. The bad guy cells work less well, so not very well, so they stop growing or decrease their growing while we're doing better to fight them off. And then we also are able to stimulate the release of these iron sequestering compounds to grab onto the iron so that they don't even have anything with which to grow. All right, so during a fever, our skin appears pale. This is due to our vasoconstriction of the blood vessels in the skin. So within the skin itself, what we're doing is we're going to constrict those blood vessels, and then we're moving the blood away from the extremities, and that's because we're keeping our core warmer. And that's in order to minimize heat loss. So I mentioned earlier, constricting or dilating the blood vessels based on temperature. And this is an example of where we see that. So when we are increasing the temperature of the body, so our hypothalamus, our thermostat has been adjusted to say, no, we don't want it to be at 98.7 or 98.6 degrees Fahrenheit. What we want to do is we want to increase the temperature. Let's increase it to 102. If we do that, that's when we're going to start to notice that we look pale. And that's because what we're doing is we are diverting the blood away from the extremities, so away from the fingers, the surface of the skin, the toes, even the cheeks and things like that will look more pale. And what we're trying to do is maintain the warmth in the core. So we're keeping the blood toward the center of the body where we have our largest vessels, which keeps it warmer. And so we can increase the temperature much more quickly if we don't have a lot of our blood in our smallest capillaries out on the tips of the fingers and tips of the toes. And so then also, not only can we keep all of the blood closest to the center and keep it hottest and increase the temperature of that blood, then also we're minimizing heat loss. And that's because the more our blood is going through all of these capillaries, the more heat is lost to the exterior as it's making its travels, particularly in the extremities because they're close to the surface of the skin and will be transferring heat from the warm part, which would be in our body, to the cooler exterior. We also have, during fever, shivering. So shivering of the muscles is stimulated by the hypothalamus. So the hypothalamus tells the muscles to shiver and to shake. And this is important because this is how we're able to, or one of the ways we're able to produce heat. And so we kind of divert our blood flow, make sure that it stays in the middle of our body so it can get to be the hottest. And then our hypothalamus tells our muscles to shake and shake and shake so that they are producing energy and increasing the temperature of the blood and increasing the temperature of the body. So when the fever breaks, as people say, our fever has broken, this is the crisis phase. This is where we have our hypothalamus is going to stimulate vasodilation. So rather than vasoconstriction, remember we're keeping it away from the extremities to keep it hottest, our hypothalamus then is going to stimulate vasodilation. So we're going to open up all those blood vessels again. We're going to spread the blood out throughout the body, not just keeping it so close to the center. This means our blood's going to return to the skin and the extremities, and then we have heat being released from the body again. So again, we have this heat transfer from the blood vessels to the exterior. The hypothalamus is going to also stimulate sweating. So this is in order to cool the skin as the sweat evaporates. So once we have this kind of switch to going from we want a fever, an increased temperature, to we no longer need this fever, our hypothalamus is going to start sweating, making us sweat, so that we can put water on the surface of the skin. And when we put water on the surface of the skin, it's much easier to transfer heat from our bloodstream into the water on the surface of the skin. And then when the water on the surface of the skin evaporates, it takes that heat with it. And therefore it cools the body because we're releasing the heat that way. So typically, as I have mentioned, a low-level fever can help overcome illness. So it's helpful, right? We talked about those three functions. So fever is good. When fever happens in yourself, your loved ones, your children, etc., it is a good thing, and we don't want to stop a fever. If we are purposely stopping a fever, then what we're doing is we're actually making it harder for our body to fight off pathogens. So if we do bring down a fever, what we're doing is we're telling the body and we're forcing that hypothalamus to go back to our 98.6 or 7 degrees. And when we do that, then our white blood cells are not as effective or efficient. 2, our pathogens aren't going to get so hot that they're going to decrease their growth. And then 3, we're not going to release those iron sequestering compounds. So we're not going to be tying up that iron and making it so that we starve our pathogens. So if we are doing the opposite of that, then essentially what we're doing is we're handicapping our body. So if we do not allow a fever to continue, then what we're doing is we're slowing down our immune system. We're handicapping our immune system, and we're giving the pathogen the upper hand here. We're bringing down the temperature to an area that's happy for them, and then we're allowing them to have that iron. So they're happy, they're fed, and they're in an environment that makes them reproduce faster and cause a larger infection. So it's important to allow fever to do its job. Our body is being smart when it goes through and it makes it feverish. However, If the immune response is too strong, then this can cause tissue and organ damage or even death. So typically, if a temperature goes up to about 104 degrees Fahrenheit, for anybody that is, I think it's age 2 and over, 104 degrees is where you're still okay. So anything below 104 degrees, that's fine. This is our low-level fever to help fight everything off. Once we reach that 104 degrees Fahrenheit, if it stays that way for more than a day, that's when we're going to start to need to go and seek out outside help, or do what you can on your own to bring that temperature down. So at 104 degrees Fahrenheit, the first time you see that on anybody, yourself or others, you're going to want to then try to bring down that fever by taking either fever reducers and or doing some cool baths or cool showers to try to bring that temperature down. Because at that 104 degree mark is when we do start to cause damage to our own cells. So our body has had that inflammatory response be too strong, and since that inflammatory response is too strong, it's going a little bit out of control, and we need to help bring it back down. If however, a person stays at that 104, if fever reducers don't help, or a cool bath, etc., then it's time to go to the doctor, time to go to a hospital, likely, because then they can put you in more professional ice baths and do some other things to help bring down the temperature, because we certainly don't want to get to a point of organ damage. So it can start to damage the brain. People can damage the brain so much where there is permanent brain damage due to fevers. And then even death. So if it gets too out of control and we have this huge response, like for example, one that we get with super antigens, then it can actually cause death. However, up to 104 degrees, you should be good to go. So super antigens. Super antigens are where we have bacterial or viral proteins, and these cause an excessive activation of our T cells. And this is from our specific adaptive response. So we'll talk more about that in Chapter 18. But it also causes an excessive release of cytokines, which we're talking about in this chapter. So again, this excessive release of cytokines. Remember, the release of cytokines is going to affect the inflammatory response. The inflammatory response is going to affect fever. So if we have this excessive release of cytokines, we have an excessive inflammatory response and we have an excessive fever. So we are overstimulating the inflammatory response. Our body is really, really trying to kill off this pathogen because it is a super antigen. And then when it does this, it goes a little bit too far. It goes overboard, and then it starts to do these things that we talked about up here. So cause some tissue damage, cause organ damage, or even death. So for example, our Staph aureus or Pyogenes can produce super antigens. So there we go. So Staph aureus and Pyogenes can produce super antigens that cause toxic shock syndrome, as well as scarlet fever. So when these bacteria get into the body, this is kind of goes along with quorum sensing. Once these bacteria get into the body, they can kind of go along doing like a normal low-level infection where our body is going to respond, have an acute inflammatory response, have a regular fever. But as they grow more and more in number, they can then produce these super antigens. When they produce these super antigens, this then can cause toxic shock syndrome. This is associated with very high, life-threatening fevers. So this is over 108 degrees Fahrenheit. Remember, we want to make sure we're taking fever reducers at 104 and keeping an eye on that so we don't go past that 24 hours or so before we go to the hospital and take care of it. If we get up to 108, this is life-threatening. So this is where we're starting to cause brain damage. This is where we're starting to have organs shut down because they're so damaged. And this is due to, again, super antigens. And we're going to talk more about how super antigens work specifically in Chapter 18, because we'll be talking about these T cells and how they work with super antigens at that time. But just at this point, note that super antigens can come from bacterial or viral proteins, or are bacterial or viral proteins from particular bacteria or virus. And they can cause excessive release of cytokines, causing that inflammatory response and fever, and then also excessive activation of T cells. And all of this leads to very high life-threatening fevers. So this is get to the hospital, and at the hospital they're going to do what they need to, including ice baths and ice tents and things like that to try to bring down that temperature so that we don't have organ failure, including brain damage and death.